Hello guys and welcome back to my channel MedPharmaco Essentials. So this is Dr. Aishwarya Vernikar and today I'm looking for a very good concept or very interesting concept I can say, that is the drug development process. So we all know that for one drug to be developed it takes around 10 to 15 years for coming up a new drug into the market. So it is very interesting topic that all the healthcare professionals should know, that is the drug development process. How much efforts does it take for a new drug to be in the market? And this can help us in the rational use of the drug by knowing how much efforts are put under for a drug to be in the market, and what all are the process that carried out during this drug development process. So let us move on to this journey or the world of the drug development process. So firstly, I would like to discuss about the phases and the stages of drug development process. So the first one is drug discovery and development. The second one is preclinical research that happens. And then you have clinical development, and then you have FDA review and post-marketing monitoring. So what happens in this drug discovery and development? So historically, drug discovery, design and development started with an identification of active ingredient from traditional medicines, or purely by chance it would be discovered in the previous times. But now there is this classical pharmacology that has come into existence, and it is used to investigate the products like small molecules. It may be natural products or plant extracts, and find those that have the therapeutic effect. Next is the steps that is involved in this stage, that is discovery and development stage. What are the steps involved? So first is we do that is target identification and validation, and next is lead identification and lead optimization. So what is this target identification and validation? Target identification finds a gene or the protein that could show its significance in the disease. So first we will identify the target here, that is nothing but the gene or the protein that shows its significance in the disease. Then later the scientist with record that targets therapeutic characteristics, they will start to record. And hence, after doing that, to validate the target, researchers use the modern techniques such as disease association, then bioactive molecules, cell-based models, signaling pathway analysis, functional analysis of the gene. This particular process has been followed by compound screening assays systems that will evaluate the effect of the new drug at cellular, molecular as well as biomedical levels. So this what is happening in this target identification. We identify the target that will show significance in the disease, and then we will validate it. Next is the lead identification and optimization. In this lead identification and optimization, the high throughput screening will be done. That is to identify the active compound or gene or any of the antibiotics that can affect the human molecule. So here we will identify that. And this lead compound discoveries are further synthesized and modified to improve the potency and reduce the side effects of that active molecule. Then is lead optimization. In lead optimization, we will conduct the experimental testings, that is using animal efficacy models and ADME models, that is absorption, distribution, metabolism and excretion tools. Then we will finally design the drug candidate. Next is preclinical research. After the drug development and discovery and development, we have the step of preclinical research. So once we found the lead compound, now what happens in the preclinical phase of the drug development begins. So in this preclinical phase of the drug development, with in vivo research we do to determine the efficacy and the safety of the drug. Here the researchers what they want to determine about the lead molecule, what they have found. That is how its absorption, distribution, metabolism and excretion information they take out. Then potential benefits and mechanism of action of that active ingredient they take out. Then best dosage they find out, an administration route that is suitable, as well as the side effects or adverse effects of that compound, and effects on gender, race or ethnicity group. then interaction with other treatment, how it can interact, then effectiveness compared to the similar kind of drugs. Okay, so all this they will determine, the researchers will determine when after once we get the lead compound here. Then in this preclinical trial or the test, what we can say, the new drug on non-human subjects for efficacy, toxicity, or its pharmacokinetic information. So we here humans are not used. Okay, it is non-human subjects we take into consideration, and these trials are conducted by scientists in vitro and in vivo with the unrestricted dosages. Here we don't particularly restrict the dose, unrestricted dosages. We see what are the results that come out in this preclinical research. Next is various assays that are carried out in this stage, that is the preclinical stage. We will discuss little. So first is in vivo, in vitro, and ex vivo assays. So in vivo preclinical researchers example are the development of the new drug using mice, rat, or models. Then and ex vivo uses animal cells or tissues from a non-living animal. Like example of ex vivo research essays are finding the effective cancer treatment agents or measurements of the tissue properties. It may be physical, thermal, electrical, optical, and realistic modeling for new surgical procedures. So in an this ex vivo assays, a cell is always used as a basis for small explant cultures that provide a dynamic, controlled, and sterile environment. Okay. So next is the silico assays. In the silico assay, they are the test systems or biological experiments that are performed on the computers or via computer stimulation. They are being assayed here. So these are expected to become increasingly popular with the ongoing improvements in computational powers and behavioral understanding of the molecular dynamics and cell biology. Next is IND submission. Once we have a particular molecule, preclinical research, once it is done, we have to submit for FDA this IND submissions. So IND is nothing but investigational new drug. So this an IND is an experimental drug that has not yet been approved for marketing by the regulatory authorities such as the FDA. But it's being tested in clinical trials to determine its safety and efficacy. So it is not yet released in the market, but yes, we are experimenting it for the safety and efficacy. The drug sponsors must submit this IND application to the FDA to conduct clinical trials on the new drug, whatever they have discovered. And IND is considered investigational because it is still under the process of investigation and its safety and efficacy have not yet been fully established here. The drug sponsors must submit an investigational new drug application to FDA to conduct clinical trials on this new drug. So before you conduct the clinical trials, IND application by the sponsors should be submitted to the FDA. The application must contain all the data of the preclinical studies such as its pharmacokinetics, dose response, formulation, toxicology studies, manufacturing information such as labeling, product methods, production, whatever methods would they have used, then batch records, all those things. Information regarding their study sites such as facilities where they have studied, equipments, what they have used, and the purpose that clinical trial protocol we say, such as what is the study design that they are going to consider, what is the endpoints, how the informed consent will be collected, and adverse event reporting, how it is done. Everything should be given in detail here. The FDA reviews the application to ensure that the proposed trials are scientifically sound and ethically conducted and that the drug is safe for the use. Okay. Next, in the drug development, IND serves as a starting point for the drug's journey towards eventual approval and marketing. So this is nothing but when you give this application, you get the permission for conducting clinical trial, and hence you may also, if it is successful, you may also get the authority to market it. Then the submission and subsequent clinical trials are necessary steps in this demonstrating the safety and efficacy of the new drug. The process is designed to ensure that the new medicine for the patient are developed and marketed safely. So for that, they take this IND application. Then in the IND, suppose if the IND, whatever you submitted, the sponsor submits the IND, and if it is approved, then the sponsors will conduct the clinical trials on patient to determine if the drug is effective and safe for the use. So now, if they get the permission, they will conduct the clinical trial. So clinical trial, there are several phases here. What we have to know first. Phase 0 is nothing but the microdosing study. In this microdosing study, what happens? The small number of healthy individuals or volunteers are being taken. That is approximately 15, not more than that they will consider. And the drug is administered at a very low dose, that is 1/10 of the human dose, to ascertain the pharmacokinetics of the drug. That is nothing but similar to that preclinical studies. And next is the phase 1. So phase 1 also we say it as human pharmacology and safety study. So here what happens, healthy volunteers around 20 to 100 have been considered, and why we do this phase 1 is to know the maximum tolerable dose and any side effects from that normal safe dosing range or what. Next is the phase 2. In phase 2, it is therapeutic exploratory. So this therapeutic exploratory, there are around 100 to 150 patients in the single center, and this is done in order to establish the therapeutic efficacy, dose ranging, and ceiling efficacy of the drug. Next is the phase 3. Phase 3 is called as therapeutic confirmatory study, where a large number of patients from several centers are being included here. Why it is done? To confirm the therapeutic efficacy of the drug, whatever therapeutic range is decided, whether it is effective or not. Then you have phase 4, that is after marketing, that is post marketing surveillance we see. Here what happens, the patient are being treated by the doctor after it is marketed, right? So here what we do is, why this is conducted, because to study the long term adverse effects and effects on the special populations like children, pregnancy, and elderly patients. So suppose now the clinical trial is done and everything is successfully completed, then you have this new drug application that is again to be submitted to the FDA. If the clinical trial successfully completes, then the sponsors must submit a new drug application, that is NDA, also known as marketing application, to the FDA for approval to market the drug in the public. Okay. Next is, this NDA are generally the final step in being drug development process. So it is a final step what you submit. Based on that, it will be decided whether you can market or stop the drug. Then NDA, whatever is given, that is the new drug application, must include all the data generated from the preclinical, clinical studies, information regarding the manufacturing process, quality control data, statistical analysis, labeling for the drug, and more. So the FDA reviews the marketing application to determine whether the drug is safe and effective for its intended use, and whether the benefits of the drug outweigh the risk, similar to IND review. So all they will review it. Once you submit this NDA, they will again review it, the FDA people. Then if the agency determines the drug is safe and effective, then it will be allowed for marketing, and hence it can be marketed further. In addition to this IND and NDA, the FDA uses other tools and purposes to assess the safety and the efficacy of the drug, including the adverse drug effect reporting, risk management plans, abbreviated new drug applications, and other types of drug approval applications. Post-marketing surveillance. So this post-marketing surveillance, we said it is the last part, where it is a practice of monitoring the safety of the pharmaceutical drug or medicinal device after it has been released into the market. So adverse drug reports any adverse effects or side effects associated with the drug use, while risk management plans aims to minimize the potential risk associated with the new drug. So about this we will deal in the next video in the detail. That is how the ADRs have been reported, how this post-marketing surveillance plays a role, and how the pharmacovigilance acts here. So thanks for watching the video. Please do like, share the video, and subscribe the channel. And I hope that the concept of the drug development process is somewhat clear by all this information.